Hey, this is Logan Gardner, and I'm going to be presenting on the effects of whey and pea protein supplementation on post-eccentric exercise muscle damage. Alright, so some background information. Skeletal muscle comprises about 40 to 45% of our total body mass. Roughly 60% of that is total body protein, and about 30 to 45% of whole body protein metabolism. The minimum protein intake for adults vary. Saturday Night Lifestyle is roughly 0.8 to 0.9 grams of protein per kilogram of body weight, and athletes are going to be higher. It's going to be roughly 1.2 to 2 grams of protein per kilogram of body weight. Some introduction. Protein supplements are frequently consumed by athletes in the belief they will enhance recovery from intense exercise by stimulating muscle protein synthesis and improving muscle damage and soreness. Most athletes already use whey protein, but over the past 2 decades, plant protein intake has increased due to environmental advantage, improved health, decreased all-cause mortality. Past and published studies and reviews have not provided the clear consensus on supplementation before, during, and following resistance exercise, and how that affects the reduced muscle damage and how it promotes muscle recovery. So the hypothesis of this study was that large doses of roughly 0.9 grams per kilogram of pea or whey protein compared to water spread throughout each day of a 5-day period would counter exercise-induced muscle damage, delayed onset muscle soreness, and performance decrements. So some considerations of past studies have been an undetermined optimal dosage. So the optimal dosage would show an increased rate of myofibrillar protein synthesis and a gains in fat-free muscle mass. Another consideration has been the strong determinant of leucine. It is questionable ideal range and dosage, but is not questionable on whether it is valuable as a BCAA. And then the ideal frequency and timing of protein. For this study, they chose the pre and post exercise and before bedtime. So this table shows the differences in amino acid composition for 100 grams of pea or whey protein, and the notable value is the leucine. So you can see that pea protein isolate has 35% less leucine content than whey protein isolate, which is a considerable amount. So for this study, this is the study design. It was a randomized trial over a 5-day period, double-blind procedure. They had 92 participants, and they had equal distribution of who got the whey protein, pea protein, and water supplementation. They compared the proteins with the water in response to exercise training. They used a 90-minute eccentric exercise protocol that consisted of 16 exercises, about 2 to 3 sets each, with 30 to 60 seconds of rest in between. And then the protein protocol was split into 3 doses per day, a total of about 0.9 grams per kilogram of body weight. And then following the exercise protocol, they had a muscle soreness rating, a blood sample, ingested another dose, followed by 4 more physical tests. Now the timeline on the left brings you through like a single day in the study. So then they showed up in the morning and they got their body composition taken in the fasted state. Right after that, they had their first dose. Following the first dose, they had a 90-minute eccentric exercise, which is the 16 exercises on the right. After the exercises, they had their blood taken immediately, soreness ratings, and then they had their second dose after that. Following the second dose, they had 4 physical fitness tests, which were the vertical jump test, leg and back strength, bench press for reps, and Wingate test. Following those, they were sent home, but given their third dose to take before bed. These are the results of the blood samples. The highlighted in red were the significant values, including creatine kinase and myoglobin, which we'll go into more specifically. But it's also important to note that there was no significant differences pre-exercise of any of the chemistry variables. So change in myoglobin. Myoglobin is a binding protein found in striated muscle tissue. It functions as a increased oxygen transportation in the muscle cell. Higher levels of serum myoglobin are associated with higher degrees of muscle damage. So when you look at the chart, you see that the whey protein is the yellow line, the pea protein is the dashed green line, and the water is the dotted blue line. So it is easy to see that the whey protein had a significant reduction of myoglobin in blood, which could be correlated to a decreased amount of damage, which would be also correlated to an increased amount of recovery, shown with the whey protein, second by the pea protein, then last by the water. So the change in creatine kinase. Creatine kinase is an enzyme found in striated muscle. It functions as a catalyst for the transfer of energy, the phosphate from ATP to creatine. It's also for storage of energy. Also, the higher levels of serum creatine kinase are associated with higher degree of muscle damage. So again, you'll see the whey protein is the yellow line, the pea protein is the dashed green, and the water is the dotted blue. You can see that around Wednesday, they started to diverge, and whey protein still had the lowest amount of muscle damage biomarkers, which would be correlated with an improved recovery or at least decreased damage. These are the results of the exercise testing. There were no significant changes due even to the significant soreness that they all felt throughout the 5 days. Participants were still able to maintain their fitness test performance throughout the 5 days. Discussion. The eccentric exercise protocol successfully induced delayed onset muscle soreness and an efflux of correlated muscle damage biomarkers for all 92 participants. Despite the soreness, they were still able to remain consistent in their performance. Whey protein was found to significantly reduce post-exercise serum creatine kinase and myoglobin biomarkers when compared to water alone. Pea protein had similar results, but the effect size was smaller compared to water. The comparison between whey and protein was an even smaller difference, but it was clear that whey had a higher reduction in post-exercise muscle damage biomarkers. Possible shortcomings of this study were that the diet outside the experiment was not recorded, and that the limited amount of time of a 5-day study gave them only smaller amounts of data. The conclusion. Although whey protein protocol of the 0.3 grams per kilogram body mass most optimally reduced the serum levels of muscle damage biomarkers, there were no influence on DOMS or exercise performance. Exercise-induced muscle damage biomarkers, creatine kinase, myoglobin, and LDH, were not strongly correlated with performance or soreness. The hypothesis was not fully supported by this data. Pea protein did not reduce post-exercise damage equally to whey protein. This may be caused by the differences in leucine content between the whey and the pea protein. In the future, studies should consider using leucine-fortified pea protein isolate supplements or greater dose volumes to match the leucine and BCAA profile of the whey protein isolate. Thank you.